Protein structure and function. In this lecture, you will identify the structure and basic chemical properties of the 20 amino acids, explain the multiple levels of organization within and between polypeptide sequences, and analyze some basic protein-protein interactions. Understanding protein structure is key to understanding function at the molecular level. All amino acids share a common structure. A central carbon atom, called the alpha carbon, is bonded to an amine group, a carboxyl group, a hydrogen atom, and a variable side chain, often referred to as the R group. At physiological pH 7, the amine group is protonated to Na and the carboxyl group is deprotonated to Cu. This zwitterionic form is crucial to amino acid behavior in solution. Amino acids exist as D and L stereoisomers, but only the L forms are used in proteins. This stereochemistry is important for biological recognition and enzyme specificity. The R group, or side chain, defines the amino acid's unique physical and chemical properties and influences how it interacts with other molecules. Side chains, or R groups, determine how amino acids behave in a protein. Similar side chains lead to similar properties, which becomes especially important when considering mutations or substitutions in protein sequences. The 20 amino acids can be grouped into 4 major categories: basic, acidic, polar but uncharged, and nonpolar. Each group influences protein structure and function in different ways. Basic amino acids include lysine, arginine, and histidine. These side chains are proton acceptors and are positively charged at pH 7. They often interact with negatively charged molecules like DNA or with acidic amino acids, forming ionic bonds important for protein folding and function. The acidic amino acids are aspartic acid and glutamic acid. Their side chains contain carboxyl groups, which are proton donors and are negatively charged at pH 7. These residues often participate in electrostatic interactions or act as catalytic sites in enzymes. Polar but uncharged amino acids include asparagine, glutamine, serine, threonine, tyrosine, and cysteine. They do not carry a net charge at pH 7, but they can form hydrogen bonds. Cysteine is especially notable because 2 cysteines can form a disulfide bond, which adds structural stability to proteins. Nonpolar amino acids include glycine, alanine, isoleucine, leucine, valine, phenylalanine, and tryptophan. These residues tend to cluster away from water, forming the hydrophobic core of proteins. Tryptophan and phenylalanine are also precursors to key neurotransmitters like dopamine and melatonin. Some nonpolar side chains have special characteristics. Methionine has a thioether group and is the first amino acid in most proteins during synthesis. Proline has a 5-membered ring that includes its alpha amino group. This structure introduces a kink or bend into the protein chain, influencing the shape of loops and turns. A peptide bond forms when the carboxyl group of one amino acid reacts with the amine group of another, releasing a molecule of water. This amide linkage creates the backbone of proteins. Peptide bonds are stable and form the primary structure of polypeptides. In the lab, this is replicated by chemical synthesis, as shown in the peptide bond formation diagram. The structure of the peptide bond was further studied by Pauling and Corey in the 1930s. They discovered that peptide bonds exhibit partial double bond character, which limits rotation around the bond and gives it a planar geometry. Peptide bonds can exist in trans or cis configurations, with the trans form being more common due to reduced steric hindrance. Polypeptides are not random chains, they have directionality. The N-terminus, or amino N, is where synthesis begins, and the C-terminus, or carboxyl end, is where it finishes. As amino acids are linked by peptide bonds, they become residues in the growing chain. When these chains exceed 50 residues, they are typically called polypeptides, while shorter ones are referred to as oligopeptides. Directionality is essential for proper folding and function. A well-known real-world application of amino acid chemistry is aspartame, a low-calorie sweetener. Aspartame is a dipeptide composed of phenylalanine and aspartic acid. While safe for most people, those with phenylketonuria PKU must avoid it because their bodies can't metabolize phenylalanine properly. This slide illustrates how understanding molecular biology helps inform dietary guidelines and medical advice. To study proteins, we first need to purify them. Chromatography is a key method for separating proteins based on different physical properties. Ion exchange chromatography separates by charge. Gel filtration chromatography separates by size. Affinity chromatography uses specific ligand-protein interactions. Each technique enables researchers to isolate specific proteins for study and experimentation. Another common lab technique is gel electrophoresis. This method separates proteins based on size and charge using an electric field through a gel matrix. The proteins move through the gel at different rates depending on their physical characteristics. A stained gel reveals distinct bands, each representing a unique protein. This technique is a staple in any molecular biology lab. To focus only on protein size, we use SDS-PAGE, short for sodium dodecyl sulfate polyacrylamide gel electrophoresis. SDS is a detergent that denatures proteins and coats them with a uniform negative charge, removing the effects of shape and native charge. As a result, proteins migrate solely according to molecular weight, allowing for precise comparison and molecular weight estimation. Proteins migrate through gels based on three factors: net charge, shape, and size. A protein's isoelectric point, or pi, is the pH at which it carries no net charge. At this peak, the protein will not migrate in an electric field. By adjusting the pH of the gel or buffer, we can influence how proteins behave during electrophoresis. Ion exchange chromatography separates proteins based on charge at a given pH. Proteins bind to a resin with the opposite charge. Positively charged proteins bind to cation exchangers. Negatively charged proteins bind to anion exchangers. Elution is achieved by altering pH or salt concentration, making this a powerful technique for protein purification. Proteins have four levels of structure. Primary structure is the amino acid sequence. Secondary structure includes alpha helices and beta sheets. Tertiary structure refers to the complete 3D folding of a single polypeptide chain. Quaternary structure arises when multiple polypeptides interact to form a single functional unit. These hierarchical levels allow complex biological function to emerge from simple sequences. Protein structure beyond the primary level is maintained by weak noncovalent bonds. These include hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals forces. Each of these forces contributes to the folding, stability, and function of secondary, tertiary, and quaternary structures. Despite their individual weakness, together they play a powerful structural role. Let's focus on hydrogen bonds, which occur when a hydrogen atom, bonded to an electronegative atom like oxygen or fluorine, is attracted to another electronegative atom. These bonds are crucial in maintaining the alpha helices and beta sheets of proteins. Figure 2.20 shows typical hydrogen bonding patterns in proteins. Next are ionic bonds, or salt bridges, which occur between positively and negatively charged side chains. These interactions are pH-sensitive and can be easily disrupted under certain conditions. Despite this, they provide strong electrostatic stabilization where appropriate in protein structures. Hydrophobic interactions are another key force. Nonpolar side chains tend to aggregate away from water, stabilizing the interior of folded proteins. Amino acids like alanine, leucine, isoleucine, and valine commonly participate in these interactions, promoting compact and functional protein cores. Finally, we consider van der Waals interactions. These are weak, short-range electrostatic attractions that occur when atoms are very close together. While individually weak, they can enhance overall stability by supporting other types of bonds in protein structures. The primary structure of a protein is the specific sequence of amino acids joined by peptide bonds. Modern techniques like mass spectrometry, including MALDI-TOF, and nucleotide sequencing allow us to determine this sequence. Although DNA can predict protein sequence, exceptions due to RNA splicing and modifications must be considered. There are notable exceptions when using nucleotide sequences to determine protein primary structure. These include alternative splicing of exons and introns, and post-translational modifications such as phosphorylation, glycosylation, and methylation. Additionally, many proteins function only when assembled into complexes with other proteins or gene products. BLAST is a computational tool used to compare biological sequences. It aligns a nucleotide or protein sequence with the NCBI database to identify similarities and homology. Different BLAST tools exist for DNA, RNA, and protein comparisons, such as BLASTon, BLAST, and BLASTX. This is an essential tool in bioinformatics and molecular biology research. Secondary structure refers to regular patterns of folding within a protein, typically involving alpha helices and beta sheets. These structures are stabilized primarily by hydrogen bonding between nearby residues. Secondary structures form the core framework for the 3D shape of proteins. The alpha helix is a right-handed spiral stabilized by intrachain hydrogen bonds. These bonds form between the carbonyl oxygen of one amino acid and the amide hydrogen 4 residues ahead. Amino acids like glycine and proline are less favorable for this structure due to their rigidity and flexibility. The alpha helix can adapt to hydrophilic or hydrophobic environments. Beta sheets are extended polypeptide strands aligned next to each other, forming a sheet-like structure. They can be parallel or antiparallel and are stabilized by hydrogen bonds between strands. This structure is common in many fibrous proteins and adds mechanical strength and stability. Turns and loops are non-repetitive regions that connect alpha helices and beta sheets. They are typically found on the protein surface, often facilitating binding or catalytic activity. These structures may form motifs, also called supersecondary structures, which are common in many proteins. Try the interactive fold game to explore these concepts in action. Tertiary structure refers to the overall three-dimensional shape of a single polypeptide chain. This structure results from interactions between side chains, such as hydrogen bonds, ionic interactions, hydrophobic packing, and disulfide bridges. It includes alpha helices, beta sheets, and connecting loops. Not all proteins fold into rigid shapes. In fact, intrinsically disordered proteins make up about 30% of eukaryotic proteins. These flexible regions still contribute to structure and function. Denaturation occurs when a protein loses its functional shape due to the disruption of weak noncovalent bonds. The protein transitions from its native state to a denatured state, losing biological activity. This process does not break covalent bonds. Some proteins are capable of renaturation, regaining function when favorable conditions are restored. Quaternary structure applies only to proteins composed of two or more polypeptide chains. These subunits can be identical or different and are held together by noncovalent interactions and sometimes disulfide bonds. The spatial arrangement of subunits determines how the protein functions as a whole. Let's compare myoglobin and hemoglobin to understand quaternary structure. Myoglobin has a single chain and stores oxygen in muscle tissue. Hemoglobin is a tetramer with two alpha and two beta subunits and is responsible for oxygen transport in the blood. Both bind oxygen using a heme group, but only hemoglobin exhibits cooperative binding, a key functional difference enabled by its quaternary structure. The quaternary structure of hemoglobin allows for cooperative oxygen binding. When one heme binds O, it increases the oxygen affinity of the remaining subunits. This is called positive cooperativity and allows hemoglobin to effectively pick up oxygen in the lungs and release it in tissues. In contrast, myoglobin, with only one chain, cannot bind cooperatively. Protein structures can be visualized in multiple representations. Ribbon diagrams highlight secondary structures like helices and sheets. Backbone models trace the path of the polypeptide chain. Space-filling models show how the protein occupies 3D space. Explore tools like the RCSB Protein Data Bank to interact with these models and analyze real protein structures in 3D. Immunoglobulin G is a great example of the domain concept in protein structure. Large polypeptides fold into independent globular units called domains, which often function on their own. Even when cleaved from the main protein, domains can retain functional activity, such as antigen binding or catalysis. This modular design is the hallmark of protein evolution. Enzymes are proteins that act as biological catalysts. They accelerate reactions by lowering the activation energy required. Enzymes bind specific substrates and convert them into products. Their activity can be measured by tracking substrate breakdown or product formation over time. Some enzyme-catalyzed reactions result in color changes, which provide an easy way to visually monitor activity. These assays are useful in both teaching and research labs to determine enzyme concentration, reaction rate, and substrate conversion. Enzymes function by lowering the activation energy of a chemical reaction. They do this through several mechanisms: stabilizing the transition state, straining existing bonds, bringing substrates into close proximity, donating or accepting protons, and forming temporary covalent bonds. These strategies allow reactions to occur rapidly under physiological conditions. Enzymes function via the enzyme-substrate ES complex. The enzyme E binds the substrate S to form ES. This complex may either return to E plus S or proceed to form product and free enzyme. At Vmax, all enzyme molecules are engaged in ES complexes, representing the maximum catalytic capacity of the enzyme. The Michaelis constant Km is a measure of an enzyme's affinity for its substrate. It is defined as the substrate concentration at which the reaction rate is half of Vmax. A low Km indicates high affinity. A high Km indicates low affinity. Km values vary significantly across enzymes and can help characterize enzyme behavior. Many enzymes undergo conformational changes upon substrate binding. This flexibility is central to catalysis. Enzymes are also subject to regulation. Allosteric effectors bind sites distinct from the active site. Enzymes may have separate catalytic and regulatory subunits, allowing cells to modulate activity precisely. Regulatory enzymes control key committed steps in metabolic pathways. They often exist as multimeric complexes with distinct subunits. These enzymes integrate positive and negative signals to turn pathways on or off as needed. Such regulation ensures metabolic balance and responsiveness to the environment. This figure illustrates how positive allosteric effectors enhance enzyme activity, shifting the activity curve to the left. In contrast, negative effectors reduce activity and shift the curve to the right. Allosteric regulation is a central feature of enzyme kinetics, ensuring fine control over cellular metabolism